All right. Good evening, ladies and gentlemen, or good afternoon, or good morning, depending on where you are. I'm very happy to welcome you in Denis and in my living room. So you can see my living room here and Denis' living room right behind him. I'm really excited to have Denis here today. He's kicking off the second series of the worldwide neuroscience talk website, I should say, platform. And before I'll give Denis the stage, I'll quickly introduce him. Denis is a doctor and a scientist, or a doctor and a doctor, I should say, at the University of Geneva. He's a neurologist and a professor looking into the genetics of neurodevelopment. Before he was a professor, he got his PhD from the University of Lausanne and his MD from the University of Zurich. He spent a little while in Boston at Harvard University and then returned to Switzerland to become a professor in Geneva. And it is worthwhile saying that Denis has just received the ERC Advanced Grant, one of the most sought-after grants on the European mainland. And we're really, or I'm really excited to have him here, and apparently so are you, because there is right now 376 attendees with another few in the waiting room. And we're all here to see you talk, Denis. So without further ado, you have the stage. One more thing, there is beautiful features in Crowdcast. You can ask questions and make polls. So if you have questions, just add them there, and then we'll vote on whether they should be asked or not. And I can call you onto the stage to ask them, or I can read them out for you, however you want. So, Denis, the floor is yours.
Okay, Tim, thanks for having me in. It's actually quite a novel experience for me, quite intimidating, I must say. I haven't been that nervous in my own living room forever, I guess. Thanks for giving me the opportunity to wear a shirt for the first time in weeks. And so, yeah, welcome everyone. I want to really thank you, Tim, not only for having me today, but for putting together this platform and for opening it to the broader neuroscience community. And as you may have noticed, there's going to be a series of such developmental events in the weeks to come, every Thursday at 5 p.m. Central European time. So without further ado, I'll be now, what should I do, Tim? Just full screen?
Something full screen? Like that? Perfect. Lovely.
Does that work?
Yeah.
Yeah, you're seeing my mouse too?
Well, show your mouse. No.
Not my mouse. My mouse.
Your pointer. I can see you.
Yeah, you're right. You're right. Okay, I'll be showing other type of mice, but you're right. Okay, it's a Francisism. Ma souris, ma flèche. Well, anyway, so it's a pleasure to be sharing some of the work we've been doing in the lab for the past couple of years or so. The talk is titled "Fate and Freedom in the Developing Neocortex." What I mean by that is I'll be trying to cover some of the aspects that control, on the one hand, the cell intrinsic, largely genetically determined aspects that control diversity of cell types in the cerebral cortex. And the freedom part relates to those aspects that either we don't understand well enough now to consider them as being deterministic, so basically aspects that we can not tie to specific genetic behaviors or input-dependent processes, but instead that might be stochastic in essence, and I call this stochasticity part, the freedom part. So work in the lab is focused on cellular neurobiology, but the questions we're interested in are those that relate to very, very broadly nature and nurture. As you know, this has been an intense area of research in neuroscience, but also in psychology, in medicine in general, even in philosophy, where the idea is to try and understand how our behaviors are determined on the one hand by genetic piece of data, and on the other hand by environmental factors. And the field has been, this question has been become quite exciting over the past few years or so with the advent of new technologies that now enable these questions to be asked and hopefully one day to be answered, not at the level only of an organism, but actually at the level of single cell types. So given a single cell type identified in the adult by a number of criteria that I will discuss in the rest of the introduction, what are the developmental factors that come into play during maturation to chisel this cell type according to cell intrinsic genetic means versus input dependent non-directly genetic means? The model system that we're using to address these questions is the mouse, and more specifically the mouse neocortex, which I assume most of you are familiar with. Very briefly, this is arguably the most complex part of the brain in cellular and circuit terms. It is a laminarly organized structure, meaning there's different layers and different types of neurons in these layers. This has been historically the first means through which distinct types of neurons have been described. And these different laminarly distinct types of neurons also differ in terms of their connectivity. Most saliently, layer 4, layer 2, 3, and layer 5 neurons can have very distinctive connectivities, whereby layer 4 neurons receive input from the thalamus, which itself relays information from the sensory organs. Layer 2, 3 neurons are intracortically projecting neurons. This would include the callosal projection neurons. And layer 5 neurons are deep layer corticofugal projecting neurons. They project below the cortex to subcortical targets. So you can see that these are very distinct cell types. You don't have to be a very experienced neuroanatomist or cellular neurobiologist to understand that a neuron whose projection stays in the cortex will need to have developmental features that are quite distinct from a neuron that will project its axon below the cortex. So these distinct features have emerged, have been sculpted by evolution. It turns out that in non-human primates and in ourselves, there's one cell type that has particularly expanded, and that is the intracortically projecting neurons. And it's thought that these cortical projections are critical to sustain our ability for abstract thinking or associational aspects between different types of sensory inputs. In the mouse, we have a 6-layered cortex, but initially, or I don't know about initially, but presumably in extant mammals, the cortex may not have been a 6-layered one and may have been closer to the one that you find in sauropsid, meaning a 3-layered cortex. So I'm spending a little bit of time on this because I think in more general terms, we won't get into that today. It's really important to distinguish what happens in the deep layers of the cortex, which are evolutionarily older, and in the more superficial parts of the cortex, where presumably one could hypothesize that some of the mechanisms that are at play here might have been recycled from older developmental mechanisms in an evo-devo kind of way. Beyond strictly genetic terms or laminar terms, of course, input itself plays a role in the differentiation of cortical neurons. There's a large body of work on this. Very briefly, just to get this idea into your mind and not be too genetically determined during the talk, there's very strong evidence that input, in particular from the sense organs, the visual cortex, or in the mouse, the visual system, eyes, or in the mouse, in the barrel, in the whisker system, input plays a critical role in setting the identity of cells, and in particular in setting the identity of the immediate postmitotic targets in the cortex, the ones that are the gateway to the cortex, the layer 4 neurons. And we have a number of papers that address this question. So what do I mean by cellular identity? I alluded to the laminar position of these neurons, which is, the most obvious one to identify in anatomical terms. More recently, over the past, 2 or 3 or 4 decades, maybe more, half a century or so, neurons have been classified, parcellated based on the expression of specific genes, or in this case proteins, which in this case happen to be mostly transcription factors. And again, you don't need to be a very experienced neuroanatomist to see that this marker here stains mostly deep layer neurons, whereas this marker here stains mostly superficial layer neurons. So this was the first hint at the fact that the laminarly distinct neurons are not only anatomically segregated, but also molecularly so. More recently, thanks to the advent of single-cell RNA sequencing, this picture of the combinatorial diversity of cortical neuron has been refined through approaches in which the combinatorial expression of a large number of genes has enabled the identification of a large number between, depending on how you lump them or split them, 10 and 50 subtypes of neurons, which are, in this particular case, distinguished by the combinatorial expression of different types of genes. And this has proven a very powerful approach and a very convenient handle to define, unbiasedly defined cell types, but also to try and look for these transcriptional signatures at earlier stages of development, with the hope to identify the sources of this extraordinary diversity. Another feature, of course, that distinguishes the different neuronal cell types is their connectivity. And historically, this is work from my postdoc in Jeff's lab, back at a time when it was still okay to drink a beer in a crowded place after work, where you would do retrograde labeling from the thalamus to look at which neurons were projecting to the thalamus, or to the opposite hemisphere to look at which neurons are projecting across the callosum. Go and look at the neurons that you're interested in after a day or two, and look at what kind of genes were expressed there. And this has proven extremely powerful to identify key transcription factors controlling the identity of these genes, of these neurons, work in particular from Jeff Max's lab, but also from Paula Arlotta's lab. More recently, we've been able to build on this to, what should I say, increase the throughput of such approaches by taking advantage of a technique developed by the Zader lab, which through a clever system of barcodes can identify the multiplex projections of single neurons. So basically can identify whether a single neuron projects to multiple targets. In a nutshell, the idea is to have a virus that you would inject in a source area, barcoded virus library, where every single virus expresses a single barcode. And this virus is transported anterogradely to any region of interest. And when you collect these regions of interest and find the same barcode, for example, here, barcode 3, barcode 3, barcode 3, it would mean that a single neuron projects to these 3 places. And so Esther Klingler in the lab has built on this technology to combine it with single-cell RNA sequencing, whereby after doing the classical MAPSIP approach, she collects the area of injections, here S1, and identifies the transcriptome of the different cells that are there, together with the barcode, allowing her to make a link between the transcriptional identity and the projectional identity of given cell types, and allowing her to identify different subtypes of intracortically projecting neurons and the associated transcriptional programs. Have a look at this reference if you're interested in knowing a bit more about that. Finally, the diversity of the cortical neurons, which, as I said at the onset, is most salient with regard to their laminar position, can also be expressed in terms of when these neurons are born. Because in the cortex, there's this interesting feature whereby the neurons that are born earliest, and you can show this by BRDU birth dating, happen to be the neurons that are the deepest in the cortex, whereas as you label these dividing cells later and later during pregnancy, you're going to be labeling more and more superficial types of neurons in the so-called inside-out pattern of corticogenesis. Okay, back to this.
Denis,
Yes?
so you know, people can see your pointer, so you might have to describe where you're looking.
Cannot
see your pointer.
Ah, damn it. Okay. All right. Should I start from the beginning? No, just kidding. Okay, so sorry about that. But do you have any feedback on whether it still was clear enough without the pointer?
There's no feedback so far in the chat, but I think.
Okay. All right.
Right.
Then, yes, so if you look here in this slide, focusing on the 12th or 13th embryonic day, what you notice is that there's a much broader dispersion of neurons in the deep layers versus the ones, the dispersion that you find at later E14, 15, or 16 developmental times. And I'll get back to this towards the end of the talk if I have time. I think there's some interesting, again, nonlinear stochastic things that are going on at these early E12, E13 developmental stages. Again, remember, this would be the time when the ancestral, the oldest parts of the cortex are being born. We'll get back to that. So the key questions, as I've alluded to, is when and how is neuronal diversity generated? And there's really two main ways of, at least two main ways through which you can think of this. The first one is one in which the diversity of the neurons here on the right is reflected by a corresponding diversity in the progenitors here on the left. So the so-called pre-mitotic, basically you look at the progenitors and you will know what kind of post-mitotic cells are there. And the post-mitotic model instead states that the diversity emerges not in the progenitors, but instead is acquired post-mitotically as the neurons are undergoing their maturational trajectories. And of course, as you can imagine, there's a little bit of both, but still understanding which process predominates dynamically during particular cortical development is really important if you want to understand, well, on the one hand, how things are set up, and on the other hand, how things might go wrong in a number of neurodevelopmental disorders. So concretely, how is this diversity generated? So in the mouse, the cortical neurons are generated between embryonic day 12.5 or 12 and embryonic day 18 or so about a week of corticogenesis. And these neurons are born from progenitors in the VZ, ventricular zone, abutting the ventricular wall, first giving rise to deep layer neurons and then more and more superficial layer neurons. Interestingly, as corticogenesis proceeds, there's another type of progenitors, intermediate progenitors here in the red pathway, which act to amplify the number and perhaps even the type of cells that are being generated, presumably accounting for the disproportionate expansion of these superficial layer neurons in higher mammals. So two types of progenitors, the apical progenitors, which you can also call radial glia, it's a more common term, and the intermediate progenitors or basal progenitors. Two axes in terms of dynamic developmental process, one that relates to the differentiation of the neurons and one that reflects to the embryonic age, meaning the time at which the neuron is born. So to conceptualize this, you can think of a matrix whereby neurons or cells in general are going to be drifting or shifting or driving themselves, depending on whether you want to be deterministic or not, along these two states, one reflecting the time at which they are born and the other reflecting their level of maturation. And by state here, we'll be talking mostly about transcriptional states, but depending on your interest, you can think of anything you want. You could think of morphology, electrophysiological properties, connectivity, etc., etc. These could be combinatorial states. So the plan of the presentation is the following. I'll be telling you about some recent work we did on how progenitors are temporally patterned during corticogenesis, meaning how they acquire successive transcriptional identities. Then some recent published work also on the plasticity of this temporal progression. To which extent are these transcriptional programs malleable and subject to be changed under certain conditions? And then I'll end, if I have time, with a little bit of unpublished work relating to basically how much of what I've told you in the two first parts actually makes sense or not. So I'm depending on that, I'll show it or not, but I don't think there's any antagonism there. So if you want to have access to, to a matrix such as this, you do want to have a technique that it gives you an exquisite temporal resolution, because you want basically to parcellate corticogenesis in space and time. And to address this, a couple of years ago, we developed this approach, which we called FlashTag, which is based on the fact that during cell cycle, the apical, I'm looking here at the left part of the slide, the apical progenitors are not static, but instead their soma moves up and down within the ventricular zone in a predictable way, such that the M phase occurs when the progenitor is touching the ventricular wall. So basically the neurons are born at that time. And I have a little movie showing that. If you focus on the arrowheads there, you just saw a cell bouncing and two other cells being born from there, basically like a basketball hitting the floor, and then two balls would come back up. And this is a relatively short event, and in the mouse, and it's thought to be about an hour, a little bit more, and is relatively constant throughout corticogenesis. So taking advantage of these properties, we injected this dye CFSE, which is an ester. We use this short name FlashTag, which diffuses intracellularly and then gets covalently bound with proteins and becomes fluorescent. And so given the short half-life of the FlashTag in the ventricle, coupled with the fact that the only cells that are touching the ventricle are cells that are dividing, we were able to label time-locked cohorts or isochronic cohorts. I'll be using that word, meaning cells born at the same time throughout corticogenesis. You can see this here on the right part of the slide too, where you see that the dividing cells in red here are initially red and also FlashTag positive. And then at 2 and 3 hours, you see these cells, the green cells, moving away from the ventricle and being replaced by a new contingent of dividing cells. The half-life of this approach is about 1 hour, probably. So when you do this, you're able basically to follow these isochronic cohorts, so meaning born at the same time, of cells on their way to the cortex. So here, 3 hours after labeling on the left, then 6 hours after labeling, 12, 24, 48 hours. What you see is that initially, I was careful to use the word cell in the title here rather than neurons, because not all dividing progenitors will give rise to neurons. A subset of them will be giving rise to themselves, meaning daughter progenitors, not quite themselves, an older version of themselves. And you can see that at the 24-hour time point, you have like 1, 2, 3, 4, 5, 6 cells or so that are still in the VZ, which correspond to cells that are cycling back into the ventricular zone and that will divide once again. So the progeny of this cell we're not taking into account, because as the cells divide, so does the FlashTag content. So if you focus on the cells that are the brightest, you're dealing almost exclusively with cells that are directly born from apical progenitors at the time you did the injection. So we took advantage of this technology to try and crack the code, the transcriptional code governing the generation of different types of progenitors and neurons during corticogenesis. And this is schematized here. What we did basically is FlashTag injections at E12, E13, E14, E15, and collected the cells right afterwards. This is the +1 hour, so this corresponds to apical progenitors. Then in another round of experiments, 1 day after the labeling, and in another round of experiments, 4 days after labeling, giving us access to a developmental transcriptional trajectory of isochronic cohorts of cells as they are differentiating. Interestingly, as cells move forward in the y-axis, meaning the axis of differentiation, they also move forward in the x-axis, such that, for example, at E14, 1 day old neurons are in a temporal environment that is similar to newborn neurons at E14. And I think these are interesting things to consider when one is thinking of non-cell autonomous interactions between cell types. So the result we got was something like this. Many of you, I'm sure, are familiar with this type of display. For those of you who aren't, this is a so-called Tisney map. You might have seen another display called UMAP, where the idea is to represent in 2-dimension an n-dimensional data set. Each one of these dots here is a cell which expresses something like 10,000 genes that we could detect, maybe a little bit less. And so this combinatorial expression of 10,000 genes has been reduced and flattened on this 2-dimensional map, where neighboring cells represent cells that have similar transcriptional programs. And what you can see, and I'm passing the details here because this is published, is that the apical progenitors, AP here, clustered together, followed by the 1-day-old neurons, the intermediate progenitors, BP, basal progenitors are here, and then the 4-day-old neurons. This is when you color code by the time of collection, so no big surprise right here. An interesting aspect, though, is now it's exactly the same graph, but color-coded by the color, you have this, what I call chronotypic organization of layout of the cells, whereby you have stripes that correspond to the time at which these cells are born, irrespectively of whether they are here neurons or progenitors, already alluding to the fact that something about when some level of transcriptional identity is transmitted from the progenitor to the post-mitotic cell. In 4-day-old neurons, this is less clear. You kind of guess some stripes, but I mean, arguably it's less clear than at 1 day, suggesting that other factors than strictly the time of birth will come into play into defining the identity of these cell types. So I'll go a little bit short on this. If you want some details again, I encourage you to have a look at the paper. I wanted to highlight some aspects that I think are interesting to many, potentially. One of the interesting things that you can do with such a data set is basically for every single cell that you have, plot it along a 2-dimensional map whereby on the x-axis you would evaluate based on computational approaches exactly when that cell is born, and on the y-axis you would compute what was the level of maturation of that cell. So basically, progenitors would be here, 1-day-old neurons here. Oh, sorry, you're not seeing it. I'm looking at, oh, damn it. I'm looking at the top left panel there in gray. So at the bottom stripe would be apical progenitors, the middle stripe would be newborn neurons, 1-day-old neurons, and the top stripe would be older neurons. And when you have such a matrix, basically what you can do, and here I'm looking below, for example, at SOX2, is for every single one of these cells to color code the level of expression of a given gene. And you can smoothen that out. Now I'm looking at the SOX2 in the bottom, and this gives you basically a typotemporal, or spatial temporal, meaning cell type and time of birth, map of gene expression across corticogenesis. So for example, SOX2 here has this corner that's darker. The bottom left corner is darker than the bottom right corner. So it's expressed in apical progenitors, but probably more strongly early on than later on. And it's not expressed in post-mitotic cells, hence the what should I call this, green color. Neuro D2, post-mitotic neuron marker, has the exact opposite distribution. Actually, I have an image here. SOX2 and Neuro D2 are like two pieces of a puzzle that you could fit into one another, and they are largely mutually exclusively expressed. CUX2, for example, a typical superficial post-mitotic layer neuron marker, is not expressed in progenitors, but instead it's expressed in post-mitotic neurons at high levels later on. And you can basically fly over this array of Greek islands, something again that we're not going to do any of us anytime soon, but so you can go on our website to have a simulation of this type of experience. and beyond, this emulation of a travel, you can actually travel within cortical neurons, and more seriously, you can use this to generate hypotheses. What do I mean by that? If you go on this website, so Jabaudon, basically what you can do is type in the name of genes of interest. So here I typed EED, which is a subunit of the PRC2 complex. And what you see here is it's a gene that's expressed at high levels early in apical progenitors. And then you have this feature which is called genes with similar dynamics. And so you can click on these genes. And then you find other genes that have exactly, or not exactly, but the closest pattern of expression to the ones you're interested in. And you can go down that tree and click to find genes that are going to be more and more remote. And I think this is potentially an interesting feature for those of you that are interested in cortical development, because if you're looking for pathways and if you're disrupted or not, you do want to assess whether the genes belonging to that pathway are expressed not only in the same cell type, but at the same time. So here the hypothesis would be that EED, DUT, NXT1, and DHFR in principle are able to interact with one another. And then you can design the experiment that you're interested in. If you were to have expression patterns that were diametrically opposite, well, if it was exactly opposite, you could argue they are mutually exclusively repressing each other. But if somehow there's no clear correlation in the expression, you would generate different types of hypotheses. So have a look and type in some of the genes you're interested in. If you want to suggest other features, we're always happy to try and oblige. The conclusion of this work, without again going too much into the details, was twofold. First, one of the striking features that I alluded to in one of the first slides of this part was that, in contrast to the classical Waddington epigenetic landscape model, whereby the diversity of neurons here schematized by the fact of a ball being in a distinct groove is generated by different differentiation trajectories. What we found instead is that the driving force for the diversity of the cell types was not post-mitotic, but was in fact related to the temporal progression of apical progenitor identity. So the model we propose is one in which the progenitors progress transcriptionally throughout time. And once a neuron becomes post-mitotic, it starts with a different ground state, but then applies a largely conserved differentiation programs that relates to the migration, the dendritogenesis, axonogenesis, synaptogenesis, etc. So the difference between adult neurons is not so much, at least in these early stages, to be found in the type of genes that are expressed here. It's the same kind of genes, but instead from the starting point from which they initiate their programs. The recipes are the same. Gülistan Acirman, who's the PhD student who led part of this work, was using the analogy of a cake that you would be baking. The recipe is always the same, whether you make a lemon cake, an apple cake, or a chocolate cake. What's different is the different ingredients that you start with. And I think it's an interesting analogy. Interestingly, for those of you who are aficionados, what we also noticed is that the identity of progenitors became more and more neuron-like as corticogenesis proceeds, which correlated well with the fact that these progenitors tend to divide more and more slowly. And the exact mechanism that underlies this is unclear, but you could imagine some level of, well, it's hard to do air quotes on a camera, but now I forget what I wanted to say. You could imagine, yeah, I forgot. Anyway, a transmission of the neuron. Oh yes, it could be contaminated. That's the word I wanted to use. So somehow progenitors that would generate successive rounds of neurons could be contaminated by some level of transcriptional programs or RNA, to put it simply, that would make them look a bit more like post-mitotic neurons. This is something interesting to investigate, I think. The second message of this study was that as corticogenesis proceeds, apical progenitors and their postmitotic cells become more and more exteroceptive, meaning more and more sensitive to the extracellular environment. We use the term extraverted here. If you looked at the type of genes that were expressed at late developmental stages versus early one, and they really related strikingly more frequently to things that are cellular sensors, as opposed to things that relate to replication and organization of the DNA. And this goes well again with the fact that these cells, basically they're in an amplification stage. They only care, I'm pushing it of course, about amplifying and making a lot of themselves, whereas here the idea is really to seemingly be able to be modulated in an environment dependent way, which brings me to the second part of the presentation, that relating to the plasticity. Well, Tim, what do you think? Does it make sense to make a question break here, or should I continue? What's the best?
Let's see what the users say, what the viewers say. I would say keep going. We've had one question whether the gene browser was with human data.
No, so the gene browser is our data, but what I didn't put here is that we were able to validate this principle, so this idea that there's a birthmark transmitted based on a beautiful data set from the Quickstein lab in the paper. Yeah.
Okay. Do you want to give me again the address of the gene browser so I can put it into the chat?
Yeah, it's genebrowser.unige.ch. Here you go. /Telagirdon. You have it?
All right. Yep, I got it.
Okay.
All right. I guess you can continue.
Okay. So the first part was about the normal temporal progression. This second part will focus on the plasticity of this temporal progression, meaning, is this successive production of different cell types by progenitors an immutable process, or is there something that can be done such that the progenitors would give rise to cells that they've either already given rise to or that they would otherwise give rise to later on? This is a long-standing area of research, and there's been a large amount of work, in particular by the Doe lab and the Diplon lab, looking at this in flies, whereby the generation of different types of neurons by neuroblasts is the result of a very well-defined series of transcription factors expressed in feedback and feed-forward loops, such that the successive expression of these transcription factors is sufficient to generate the cell type. So the consensus here, the key word here is motoneuron competence, is that as cell divisions occur, the competence of progenitors to generate cell types becomes more and more restricted. At the beginning, you can generate a lot of things, but as you generate things, you can not generate what you've previously given birth to. In mammals, this seemed to be the case too, largely stemming from a beautiful series of experiments from the McConnell lab in the 90s or so, done in the ferret. And I'm going to drive you through these experiments. Since you don't see my pointer, I'll drive you through this in words. So what you're seeing here is, on the left, in blue, the brain of a young embryo is supposed to be ferret. So you should imagine this being gyrencephalic, but that's a detail. And the older embryo is on the right in red. So in a young embryo, that's in the bottom left now, deep layer neurons are normally being produced, whereas in the older embryo, superficial layer neurons are being produced. Abbreviations here: DL, deep layer; SL, superficial layer. So what Sue McConnell and her team did was first to take progenitors from young embryos and transplant them into an older embryo. And when this was done, the progeny of these cells was superficial layer neurons instead of deep layer neurons, showing that, consistent with the fact that early born neuron progenitors are pluripotent and can anticipate the type of neurons they're going to do. In a way, it's not strictly speaking a plastic effect. You're doing something that you would have done otherwise, but you're doing it early than you would. So it doesn't necessarily imply a changing of the transcriptional programs per se, but it implies a change in the speed of these transcriptional programs. And in contrast, when the reverse experiment was done, apical progenitors being transplanted, so apical progenitors meaning radial glia, presumably, transplanted into younger embryos. Instead, there was no plasticity, and these progenitors kept on generating cells that they would otherwise have generated in their natural host. Again, exemplifying this progressive restriction in fate. You cannot do what you've already done. So this is the so-called ratchet mechanism of progression of cell fate. The wheel only turns one way once you click past a threshold, whatever that would be, an epigenetic threshold. You cannot go back. However, this somehow is in contrast to a large body of literature showing that if you express single transcription factors, or for that matter, historical experiment by Gurdon, if you transplant young nuclei or more totipotent nuclei, or in this case differentiated nuclei, and put them in another environment, you can generate things that have already been generated. So it seems like the arc of development is not something immutable, but instead that, given the proper circumstances, cells can be reversed to previous states. So we wanted to try and understand this with a little bit of a better cellular resolution and with the genetic tools that are available in the mouse. And to do this, Paulina Oberst, a postdoc, a former PhD student in the lab, now a postdoc in the Studer lab in New York City, did a series of experiments basically trying to replicate these seminal heterochromic transplantation experiments. So the setup is quite simple. At E12.5, I'm looking at the left here, progenitors give rise to deep layer neurons. At E15.5, they give rise to superficial layer neurons. The question is, what happens if you take E15 progenitors, here in red, and put them in an E12 environment? So heterochronic backwards transplantation. The way Paulina did this is using flash tag, collecting cells immediately after the labeling, fact sorting them, and she used 3 types of settings: isochronic transplantation from E15 to E15, so late into late; isochronic young into young, 12-12, and then the interesting experimental one, the 15 into 12. So I'm going to use this color code throughout. Pink is late, blue is early, and purple is the experiment. When we do this, when she did that, she had essentially progenitors. They were SOX2 positive and did not express either the intermediate progenitor TBR2 marker, TBR2 eomes, or neuronal markers. So we're dealing with a population of largely apical progenitors. Immediately after transplantation, I'm looking at the bottom left here, she could see that the cells integrate, divide, perhaps to some extent, and then migrate up and up. This migration is quantified here on the far right and is compared to the type of migration that you see with a flash tag injection. And by comparing the position of these different boxes, you can tell that these apical progenitors, radioglia, integrate, or the progeny of these cells migrates at a normal rate. You can identify cell divisions, and you can very rapidly, in fact, and this has been previously described, identify radial glia-like processes. I don't know if this is showing well on your screens. as soon as 24 hours after the labeling. I didn't mention, of course, that the cells that she's transplanting are RFP positive, whereas the host is not RFP positive. So this enables her to identify the donor cells. Interestingly, the cells would integrate into very specific loci. So there was a little spots where these cells would integrate, and allowing her to define pseudo clones or oligo clones, because if you look early on, you only have typically 1 or 2 cells per each integration site, such that looking at later developmental time point, if you see a number of cells, you could assume that they were born from a limited number of cells. We'll get back to that. So what are the data? 15-15 isochronic transplantations, the cells end up in superficial layers, no big surprise. 12-12 isochronic, so obviously initially the cells that are being generated are the deep layer neurons, but then as time goes by, this is collected at P7, these E12 progenitors are going to give rise to superficial layer neurons, so you have cells both in deep and superficial layers. And the question now is what's going to happen in the E15-12? Are we going to have a pattern where cells are essentially exclusively in the superficial layers or throughout all layers? And the answer is here. Indeed, the distribution of the daughter cells in the heterochronic 15-12 condition corresponds to the one in the isochronic 12-12, demonstrating a change in the neurogenic potential. So you can quantify this in a number of ways. I'll stick to this one for this presentation. Basically, what you have here is superficial layer and deep layer and the fraction of cells in each of these layers. So 15-15, essentially all the cells are in the superficial layers. 12-12, it's 50-50. And you see that 15-12, it's 50-52. So throughout the next slides, I mean, for sake of simplicity, the question is really whether the purple is going to look like more like the blue or the red. Spoiler alert, in most of the cases, I mean, in essentially all of the cases, the purple and the blue are indistinguishable. Laminar identity, so that's one case solved. Is the molecular identity of the daughter neurons reascribed? So we use this, we check this using a number of markers. Here, COX-1, a superficial layer marker, and looked at the molecular identity of the cells in the superficial layers, and you can look at the bar graph here on the far right, whereby, of course, in the 15-15 condition, most neurons are expressing superficial layer markers, and the deep and the early stage, it's only half of them, and the identity is appropriately reascribed in the heterochronic transplantation. One thing I want to draw your attention to, if you look at the AP 15-12 in the 1, 2, 3, 4th rectangle from the left, the cells that express COX-2 are the ones that are located in superficial layers. So it's not like they randomly express this marker. Sorry, I said COX-2, I meant COX-1. The cells that express COX-1 are expressed at their proper place. And you can do this with a number of markers here, the deep layer neuron marker CTIP2, layer 5, and another layer 6 marker TBR1. So the molecular identity is appropriately reascribed in these cells. Keep in mind that in principle, we'll get back to that, you could argue that this might not reflect a reprogramming of the progenitor, but instead a post-mitotic reprogramming of the cells based on where they are in the cortex. Connectivity-wise, again, the cells behaved like the daughter cells of early-born neurons. I'm looking here at the E15-15 condition, where in the top square, this reflects retrograde labeling from the callosum, and in the bottom square, this reflects retrograde labeling from deep cortical structures. So it's all white, it's normal, so it means that most superficial layer neurons never project to the deep layers. And now if you go to the far right of this box, you can see that there's the presence of subcortical projections in normally late-born neurons when you put them in a young environment, demonstrating cumulatively here reassignment of the laminar, molecular, and circuit axonal projection identity of these cells. So what's going on? mechanistically speaking? Are we dealing with something that's happening at the level of the progenitor, or is it post-mitotic? So to try and understand this, we looked at the molecular identity of the progenitor, their membrane potential, their neurogenic properties, and their clone size. And I'm going to speed up a little bit because I'm a little bit late on time. In a nutshell, the molecular identity of these progenitors was reascribed and reassigned to that of normally deep layer neurons, and we're going to talk about one of these genes in a few, one of the genes that are induced in a few minutes. The membrane potential of the progenitors was reassigned. We showed 2 years ago now that early on in corticogenesis, the progenitors are more depolarized than later on, and this was also the case for the progenitors. And I'll go fast on the neurogenic properties of these progenitors very briefly. Just look at the blue and the purple bars, which are at the same level, indicating that there's a plasticity in terms of self-replication and neurogenic properties. And finally, the clone size, of course, early-born neurons, progenitors that are early in corticogenesis have more time to give rise to neurons, so they have a bigger clone size than the early ones. So again, here in the transplanted cells, we found that a larger number of cells were present in each of these oligoclones that I introduced to you before. So what's going on? How can we reconcile our findings with those of the data in the ferret, where there was strictly no plasticity when late-born neurons were transplanted into the younger host? Well, we think what this reflects is the fact that the germinal zones of these two species are very different from one another, whereas in the mouse, the progenitors are born from the ventricular zone and the subventricular zone. In the ferret, instead, there's this very expanded region called the inner and outer subventricular zone, which predominates at later developmental time points. And given the way through which these cells were collected at the time, and it was a BRDU pulse labeling, what we think might have happened is that a large fraction of the cells that were collected in the heterochronic experiments back then were in fact intermediate progenitors, which presumably would be more fate-restricted than a genuine radial glia cell. So to test this, well, we replicated the experiments in the mouse using BRDU birth dating, but also here using flash tag. The idea is to do the flash tag labeling, wait 10 hours for the cells to become intermediate progenitors, and you can see that we have about 80% of the cells that are TBR2 positive, and do the exact same thing than what we've done. And what you can see here is that indeed, oh yeah, this is the data for the BRDU, but you can have the same, there's the same data for the intermediate progenitors, is that in contrast to the apical progenitors where you have a 50-50 different, in contrast to when you use the flash tag in the apical progenitors where you have a 50-50 distribution, the plasticity is lost in the intermediate progenitors, strongly suggesting that the lack of plasticity seen in previous experiment reflects not a lack in the ability to generate previous cell types, but instead the fact that the cells that were transplanted there were already fate-restricted cells. So how does that work? What's the molecular mechanism underlying this? Very briefly, we focused our attention on the Wnt signaling pathway because Wnt7b in particular is expressed at high levels in neurons early in corticogenesis, and we had in the transcriptional data elements that would suggest that the Wnt pathway would be involved. So what we did basically is to investigate this possibility in vitro. We isolated apical progenitors and looked at their neuronal output, defining superficial layers as CTIP2 negative and deep layer neurons as CTIP2 positive. And we manipulated Wnt signaling either by activation or repression. So I'm going to drive you through this. If you do this in vitro at E12, using E12 progenitors, you're going to generate about 25% of deep layer-like neurons. Of course, you're generating superficial type neurons at some point. If you do it at E15, you can look either at the pie chart or at the bar graph here. You have the smaller fractions of such cells being generated. If you now activate Wnt signaling, you're going to be generating, so as is the case during early to levels corresponding, presumably, to the ones that are high in early corticogenesis, you're going to be generating a larger fraction of deep layer neuron-like. If you co-culture old apical progenitors with a young cortex, basically you can replicate this effect, suggesting that something is secreted by the young cortex to change the fate, the neurogenic potential of progenitors. And basically you can block this effect by blocking Wnt signaling in the apical progenitors. So demonstrating directly that the element, which is the substance which is released by the young cortex, is indeed acting on the Wnt signaling pathways. And we confirm this in vivo by blocking the Wnt signaling pathways in the cells that we transplanted using a dominant negative form of TCF4. And you can see here in the middle panel that in contrast to the control condition where you have cells that are respecified into deep layers, this plasticity is lost if you block Wnt signaling. So in a nutshell, with regard to this second part of plasticity, the message I want to give here is that it's important to not only think of potential in terms of when during, in this case, corticogenesis, the cells are being collected, but instead that there's a cell type specificity to the ability of cells being to be reprogrammed. So of course, this is a very unnatural setting because it doesn't happen often in nature that you would have to find a way to survive when you're transplanted into a younger animal. I don't think there's a lot of evolutionary pressure for this kind of property, but I think it's an interesting, laboratory model with regard to the potential of cells. And so with this, I think I'm going to stop here. Maybe depending on where the discussion goes, I could show some elements of data of this last part. If you're interested in stochasticity, I would just say, have a look at this nice paper by the Marine Lab, which came out recently, and a comment by Esther and myself relating to this paper. And within a few weeks' time, well, hopefully days or weeks, we will be posting on bioarchives a manuscript that relates to this third part that I won't have to go through, but basically which relates to, let me just show you one image. Here you go, to the fact that at early developmental stages, there's a broad diversity in the laminar molecular circuit identity of cells born at the same time, whereas this is much less so the case later on in corticogenesis. And we think this reflects the progressive implementation of differentiation, of control programs controlling the fidelity of neuronal differentiation during corticogenesis. So this will come out, at least be posted on bioarchives in a week or two. All right, so with this, I'd like to thank the people who did the work, and in particular, Gülistan and Ludovic. Ludovic is now an assistant professor at the University of Lausanne, and Gülistan was a postdoc in, a PhD student in the lab. Palina is the first author on the second part of the story in the plasticity, and Elia has driven the part relating to the thing that I haven't shown you, but that will be available soon. So thank you for your attention, and I'm happy to take questions if there are any.
I'll clap for everyone, I suppose. You do have questions. I invited users that have questions that got voted up onto the screen if they want to ask them themselves. If not, you can look at the questions maybe in the bottom of the screen.
So I click on ask a question?
Yep.
And I take them like that. Okay, oh yeah, that's nice, okay. So the first question from Eric is, what happens if you transplant 12 into 15? Yeah, that's a good question. I have a little schematic for that. Where is it? Here you go. Are you seeing this?
Yes, we are.
Okay. So normally on the left, this is the normal corticogenic progression from deep to superficial layer. What happens, and you can look at the data in the paper, it's in the supplemental, but I think it's quite detailed, and I hope you'll find it convincing. Initially, the E12 cells, so transplanted at E15, are going to give rise to deep layer neurons. So basically, they're not plastic. But the funny thing is, starting from E17, 18 or so, the neurons have a migration defect, probably because there's not as much radial glia, and maybe for some reason the extracellular matrix becomes less permissive, such that you have an outside-in construction of the cortex. Basically, cells being later born get stuck deeper and deeper. And again, have a look, but I think we've shown it quite convincingly. You can show this with the BRDU pulses. So it gets a bit complicated, but I think these two first steps suggest that you don't have a plasticity. And again, I want to emphasize that to me, anyway, if you had it, it wouldn't be plasticity per se. It's an anticipation of a program that already exists. And to me, it also makes sense that these cells would be less prone to be plastic because of their transcriptional nature, which is not caring that much about the environment, but instead being focused on dividing. But this is a bit reductionist, I agree.
Should I just read the next question to you, or do you want to read
I have it there, yeah. Hi, the radial glia cell also generates oligodendrocyte and astrocytes. Yeah, if you do the transplantation, do you alter the glia number? I don't know, but what I know is that if we do it later on, meaning if I remember well, E17, we take E17 progenitors and do the transplantation into a younger animal, they remain gliogenic. So meaning that this plasticity is restricted to the neurogenic period of corticogenesis, which is actually quite interesting. Why would they not be plastic when they start making glia? I have no idea. Yeah, I don't know. How do you reconcile the finding by Uli Müller's group on early-born supragranular neurons with your data? Yeah, so for those of you who are not familiar with this, there's data from Uli's lab, which suggests that there's fate-committed progenitors even early on in corticogenesis, meaning cells that are poised to make late-born neurons, superficial layer neurons, but early in corticogenesis, and presumably they would be quiescent or dormant. So two possibilities. One would be that we're not labeling them with the flash tag because for some reason if they're not mitotic, we would not hit them because of the nature of the labeling. The other possibility is that, as you know, these are controversial findings, and it's unclear that there's indeed COX-2 positive progenitors in the ventricular zone. We never saw them in our own data. So my own position would be to say, if they exist, they're not very frequent. Now, you could generalize your question as to the existence of fate-restricted progenitors in general. The Yorka data alluded to suggest that there's at least 3 types. Yeah, maybe, I don't know. I think it's a question of power resolution. How many cells you need to look at to find a rare cell type. They might be there, but for me right now, yeah, I remain agnostic with regard to this question. I don't think they're there, but I'm happy to be convinced otherwise. If apical divisions, another question, if apical divisions are asymmetric, how almost all cells become IPs after 10 hours? Yeah. Well, that's not incompatible to my knowledge. I mean, the question, I guess, is, I guess Alex is surprised by the fact that at E15, when we do the flash tag and wait 10 hours, we have so many TBR2 positives. Yeah. I think some of them, I mean, it's not I think, I know some of them are in fact neurons because we can identify them in the host. The way we do that is we chronically give BRDU in the host, and we look at cells that are BRDU negative. And these transplanted neurons, baby neurons, right, they're 10-hour-old, they're not plastic. So they go to the layer they're supposed to go to. So yes, I guess that's the best answer I could give at this stage. If you have something more precise, just send me a mail or we can chat. Regarding the browser, oh yeah, do you get, so this we answered. Could it be possible that IPCs at E15 suppress APs from going back? Well, I don't think so, because the apical progenitors, they are able to go back in time. So I don't think there's any evidence for that. Oh, this has moved now. Oh, here you go. Isn't the Wnt effect on E15 transplanted progenitors in E12 at odd with the fact that E12? Yeah, that's a good question. Yeah, that's a really good question. Yeah, I don't know what to answer there. Let me finish the question.
Can you read the full question, because it's not, we often can't quite see which one you're reading, so it's good if you.
Oh, sorry. So I'll read the question. Isn't the Wnt effect on E15 transplanted progenitors at E12 at odd with the fact that E12 progenitors are supposed to be not very sensitive to extracellular signals? So Isabel is saying, yeah, I mean, if on the one hand, these progenitors are not sensitive to extracellular signal, why would they need to have so much Wnt in the cortex to start with? Yeah. I don't know. Again, I mean, the picture is not black and white. The cells become more exteroceptive as corticogenesis unfolds. You know as well as I do that cell-cell interactions also play a role in setting neurogenic properties early in corticogenesis. I'm not an expert on Wnt signaling in the cortex. I think there's data from the Tarabikin lab, and of course Shubatole had some work, but not in the cortex. Those of you who are more knowledgeable than I am on the effect of Wnt probably can find other answers. But yes, I agree, there's some level of paradox. Then the data from your last experiment suggested that 80% of flash tag labeled progenitors are TBR2 positive after 10 hours. Is it your assumption that the remaining 20 are terminally dividing? Okay, so this dividing RGCs or BIPs, i.e. do all progenitors destined to become neurons at some stage become TBR2 positive expressing? That kind of relates to the previous question. The answer is I don't know, and I don't think there's very good data on that. The problem with TBR2 is that, as you might know, it's transiently, at least transiently expressed also in postmitotic cells. So if by immuno you want to identify IPs, you do want to take TBR2 and Ki67. So I'll remain agnostic on this one too. But no, I guess this approach is not the best one to answer these kind of questions. The cells that you transplant only integrate in the neocortex. Yes, Virginia, that's a good question. The answer is no. Since Palina injected in the ventricular space, in some cases, the progenitors would integrate at other places. Well, not in some cases, but in most cases. In other places, including the ventral pallium and other places. And it seems from data that's now ongoing in the lab that the progeny of these cells look a lot like what they're supposed to be when they're born from there. So we're talking about spatial plasticity there. So this needs to be confirmed. But again, if you think that it's a parsimonious mechanism to change the identity of the progenitors while keeping the differentiation path of the neurons to generate diversity, you could argue that it's a parsimonious mechanism to have a constant progenitor identity, but then that would be modulated based on space. And actually, there's a cool paper that came out today on bioarchives by Guillermina Lopez Bendito's lab, showing that there's region-specific transcriptional signatures across brain regions. So if you look at glia or neurons in the thalamus versus in the cortex, they share common signatures that are cell type independent to some extent. And if you reprogram an astrocyte into a neuron, and you do that in the thalamus, you make a thalamic neuron, and if you do it in the cortex, it's a cortical neuron. So I think the plasticity of apical progenitors and neurons is underestimated and will, to a large extent, depend on the niche. It seems that environment influences E15 progenitors when transplanted at E12, yes. How Wnt pathways affect molecular mechanisms and modify cell fate? Yeah, so Robert wants to know exactly the molecular pathways. I don't know. This is an interesting, we don't have the cellular resolution, the molecular resolution with the patch-seq to go into the details of exactly how that would work. We know, you're asking epigenetics, we know that the PRC2 complex is involved in setting the tempo for the progression in time and in temporal identity. And EZH2, which is a subunit of that complex, was modulated and has been reported to be modulated by Wnt in the retina. So you're not seeing it, but I could be hand waving a little bit, but it's possible. How might extracortical afferents factor in the initiation or maintenance of neocortical cell fates, thalamocortical on layer 4? Yes, so that's a long-standing love of the lab, trying to understand how input-dependent factors act to drive or to modulate identity. I would refer you, for example, to the paper Pouchelon et al., Nature 2014 or so. There's also a lot of work from Denis O'Leary's lab. It's worth a discussion in itself. Unfortunately, we don't have a happy hour right after, but have a look at those papers, and I'm happy to answer your questions if you have any. And then finally, not fine, man, they're popping up like.
Yeah, there's a few more. There's if you go scroll.
Okay. It's okay. I'll go back up after. What are the intrinsic differences that define early and late progenitors? I guess. Oh, this is from Nicoletta. Hi. Intrinsic. You mean genetic differences, or you're asking amongst the temporal programs? What's nice is that I can interpret the question any way I want, right? So anyway, among these temporal programs, which ones are actually cell intrinsic versus driven by the environment? Yes. A viewer asked us that question, and at first we were upset, but I thought he or she was right. There's some data in the supplemental where we basically, let me not say something stupid. What did we do? Yeah, we isolated the early progenitors and then the late ones and looked at their neurogenic competence in vitro, and saw that it was conserved in line with the data of the Van der Regen lab. So at least for the crude readout of CTIP2 expression, it seems to be cell intrinsic in terms of neurogenic competence. Now, whether the detailed temporal programs are conserved or not, I just don't know. It would be interesting. Indeed, it's hard to do because the thing is, they integrate very sparsely, so it's an advantage because you can then have this pseudoclonal or oligoclonal analysis, but it doesn't. It would be nice to have a cluster of late progenitors in an early brain and see what happens. Yeah, I don't know how to do that efficiently. Relate, so I go back, here, 0 votes. Okay, I'll go back from the top. Does the connectivity morphology of transplanted neurons change relative to their original partner? Connectivity, I don't think we did it for that paper, but I would bet they do. I would be very surprised if they don't. Well, connectivity, yes, we did it. We looked at the axonal projections, and yes, it's reascribed. Is anything known, but we don't have it with a cellular resolution, meaning you're talking about their original partners, but they go to a different place. Is anything known about whether the Wnt mechanism or the pathway is involved in how does it regulate? So I guess this relates to the question we had before. I don't know in detail, but perhaps PRC2 and epigenetics. Regarding the browser, okay, this we had. Can you speculate on the extracellular signals that act upon the transplanted E15? Well, we did more than speculate. It was the Wnt the Wnt7b. So have a look at the paper. So we think it's secreted from the cortex. I mean, we don't think. I think there's good evidence that it's secreted from the cortex at E12. Related to your introduction, the circuitry being input 4, 2, 3 to 5 output, how can we explain the temporal generation goes 6, 5, 4 to 3? Yeah, that's a good question. That's actually an excellent question. So basically, meaning since layer 5 is the like how, I guess it makes sense in evolutionary terms, right? Because layer 2, 3 is born after layer 5, so they would know that layer 5 exists. I mean, I'm being very reductionist here. They have to pass by layer 5. The question would be layer 4 to 2, 3. You're saying, how could layer 4 connect to 2, 3 if at some point there's no layer 2, 3? There's an intimate relationship between layer 4 and 2, 3. To me, they're the same cell type. There's nice data from Marta Nieto's lab showing that initially during development, layer 4 neurons have axonal projections that they then retract. So the logic, I think, is kind of straightforward. The functional advantage, I don't really know what to say. I mean, for me, this question is interesting because it asks the question of what are the spatial constraints over the establishment of a specific type of circuit? And we're looking at this in the lab by looking at this type of connectivity in vitro. So again, back to this intrinsic and extrinsic controls. Do you have examples on how exteroceptivization, of progenitors over time affects the properties, genetic programs of the mature neurons, Yeah, I don't know if it answers your question, but the fact to me that these progenitors become more exteroceptive is another way of saying that they become more neuron-like. They're more excitable too. And this relates to one of the things I was showing on the Waddington landscape of the fact that these progenitors become more neuron-like as time goes. So examples of, yeah, I guess I'll remain fuzzy on that one. I think as time goes by, progenitors become more like their daughter cells. They're closer and closer, and eventually they get stuck in a limbo, which is called astrocytes. Now, this last part is a joke, but still, you can think of an astrocyte as a low-level replicating progenitors. Although I don't think it's a continuum in terms of plasticity. I think it's an interesting work for future studies. Okay, last one. Oh, two more. Could Wnt, I have to go home soon, so no, I'm just joking. Could Wnt have this effect indirectly, just affecting the cell cycle time? Yes. Wnt activation would mean faster cell cycle that corresponds to younger progenitors. Yeah, I don't know. It's a very good question. The relationship between cell cycle length and competence is, to me, an interesting topic. There's a number of people that are looking at that. Debbie Silver had some nice work a couple of years ago, and Colette Dehaye also, Franck Polleux at the time. I personally think that cell cycle is a determinant, cell cycle length is a determinant of the type of neurons that will be produced. It makes sense if you're thinking of, like, for example, transcription factors that would have a low affinity of binding for a given site. If that particular site is available for longer, then the probability that it would bind would be higher. So you could actually change properties without a need for a transcriptional, salient transcriptional changes. So yes, I would be interested in finding, I mean, I'm sure there's literature out there on link between Wnt and this cell cycle and then fate, but I'm not familiar with it. Do you think those neuron-like progenitors would be different from the gliogenic ones? Haha, yes, I don't know. It's the question as what is the continuity between, the lineage continuity between astrocytes and neurons? No data from, in particular, Seymour Hippenmeyer and Songhai Shi's lab relating to the MADAM, where these clones have astrocytes towards the end, suggesting that at least some progenitors can give rise to both neurons and glia. I don't know. Do you think I'll take a pass? I don't know. Could wind have this effect indirectly? Just, oh, that's the same question than before. And then, yeah, good question from Alain Chedetal. I guess hairdressers are closed in Switzerland during the lockdown. They are, but actually they are going to be the first one to open with the doctors on the 26th. I did shave, you noticed, and yes, I'll get a haircut. I welcome any suggestion on this. I'll take a look at some of your pictures and try to inspire myself, as always. Thanks for the question, Anna. Okay. So on my side, that's it. I don't know, Tim, what would you want
to... Or no? Do you think neuron-like progenitors would be different from gliogenic ones?
Yeah, this we had. I already answered, Isabel.
Okay, sorry. All right, I guess that's it then.
I need to do.
All right, so thank you everyone for being here. Next week I'll be in next week I'll be in Tim's sofa and chairing from there. And our guest next week is going to be, if I'm not mistaken, Franck Poileux. Is that it? Yes, Franck Poileux. 5 p.m. So looking forward to, if not seeing, at least reading you there. And stay safe until then. Take care and have a good weekend and week.
Thank you, Denis. I suppose now we wait until everyone logs off.
So we're out now. We can. We're not online.
No, we are, but I don't know that.
So I shouldn't like touch my nose or.
No, try not to.